In this video, we will discuss about the interplay between E-Cadherin and Wnt signaling. Although this Wnt signaling is the most conserved signaling pathways within the cells, um, but its aberrant activations might cause the cells to acquire the cancer cells-like properties like uncontrolled growth rate and the the more survival probabilities. So how these things are related? So as we have discussed in the previous videos about this E-Cadherins, intercellular signaling pathways that once the E-Cadherins interact with the other E-Cadherins, it will trigger a complex formations in the intercellular parts that composed of alpha, beta-catenin and P120 Cas. And what will happen in this case is that the beta-catenin is being consumed mostly in this E-Cadherins intercellular signaling pathways activations. However, the beta-catenin also plays a role in these Wnt signaling pathways. So how does it plays the role? Is that when the first we discuss about what happens with this free beta-catenin. So this free beta-catenin that are the leftover beta-catenins from this inside the cells after this E-Cadherins intercellular part complex formations, it will interact with the APC, and also thereafter it will interact with this GSK, 3-beta, conductin and axin to help in the phosphorylations of this beta-catenin. And this once the phosphorylation of this beta-catenin occurs, it will then direct it toward this ubiquitination for its degradations. However, when the Wnt signaling pathways are activated, it will prevent the phosphorylations of this beta-catenin, and thereby most of the beta-catenins will be left in the cytosols as a free forms, and this free beta-catenin will then move into the nucleus, and thereby it will upregulates these TCF transcription factors. TCF transcription factors are directly related to these Wnt signaling pathways. That is, it will helps the cells in terms of the more survival rate or high proliferation rate, and thereby, so in the case of the cancerous cells, when the Wnt is aberrantly activated, then it will cause the high free beta-catenin within the cytosols, and this will high beta-catenin in the cytosols will cause them to move into the nucleus, and where it will upregulates the genes that are controlled by this TCF transcription factors, and these genes are then responsible for the uncontrolled growth rate or high proliferation rate of these cells, and which is one of the characteristics of the tumor cells where they want to grow rapidly, and in order to do so, one of the approach is to activate this Wnt signaling pathways, and also so its reverse is also could be true, that once the higher beta-catenins are present within the cells, then it might cause less E-Cadherins interactions, because beta-catenins are present in the free form, so they're not able, it means that they're not forming a complex in the intercellular part, and thereby will also help the beta-catenin to promote the high proliferation growth rate. The other things that they have mentioned about here is this tyrosine kinase. So in this tyrosine kinase, what will happen is that once it gets activated, it will form a it will cause the phosphorylations, and that will also promote these complex formations, or we can say that tyrosine kinase will also interact or take place a role or play, or this beta-catenin also plays a role towards this SRC or the tyrosine kinase activations. Once the cell surface, this EGF and HGF gets activated, then intercellular part tyrosine kinase will also requires this beta-catenin to make a complex. and thereby this makes this beta-catenins interplay with the WNT, E-Cadherins, and also to some extent with tyrosine kinase, the complex interactions which if gets disrupted might change the cells towards behaving like a cancerous cells, that is high proliferations and survival rate. So that's all for this video. I hope you understood briefly about the E-Cadherin and WNT signaling interplay. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.